Hi there. Nice to virtually see everyone. This is Olav Hellebo. I'm CEO of ReNeuron, and like to give you an update on our progress. So we're a public company listed in the UK on the AIM market. So here's the disclaimer for that. And just quickly the highlights. So we're obviously a cell therapy company. In the UK, we have our clinical team, or parts of our clinical team sitting in the US, and we're working on a RENE.L and a neural stem cell platform. The lead program is in Retinitis Pigmentosa, which is an orphan drug area. We have fast track designation in that program, and we have a phase 2A clinical trial that's ongoing, and I'll update you on the progress on that in a few pages. Following that, our expectation is then to do a single phase 3 study starting second half 2022. We have some very interesting research programs ongoing as well in exosomes and induced pluripotent stem cells, so I'll cover that briefly. We're well funded after a recent capital increase. We raised £17.5 million before Christmas, so that gives us the opportunity to cover pretty extensive news flow over the next 18 months or so. So these are the 4 platforms we're working on. The key one is the human retinal progenitor cells. So these are delivered subretinally. We believe that's the right way to do this because it enables engraftment, and that's always been our approach. We think that is the best approach for both a profound and durable efficacy. We've been able to cryopreserve these cells, which is obviously important, not only for clinical trials, but also later on for commercial launch. So this allows really global ship, store, and use anywhere in the world. The data we have so far in the phase 2A is very positive, and we've decided to expand that study, and I'll take you through that in a bit. We are unencumbered except in China, where we have a partner with Fosun Pharma, which is one of the big companies in China. The exosome platform is using our neural stem cell as the starting point. We have high-yielding exosomes being produced. We're focused on loading these exosomes with different cargo gene therapies, particularly looking for delivering across the blood-brain barrier, which is obviously a big need, and we have several pharma collaborations ongoing to do that. The third leg is iPSCs. So we've used our CTX cell to go backwards and create a pluripotent stem cell platform where we've been able to preserve the immortalization of the CTX cell, which then gives us a potential here to create allogeneic cells of a multitude of different kinds of cells, CAR-Ts or pancreatic islet cells, for example, for production of insulin. So this is where we're working these days. The CTX cells is no longer being pursued by us, but it's open for partnering, and is partnered with Fosun Pharma for China. The next milestones, I mentioned them quickly. So for the human retinal progenitor cells, it is continuing our current phase 2A study and then getting into a phase 3 study after that. So let me go a little bit more in detail through that program. So retinitis pigmentosa is inherited degenerative eye disease. The incidence is about 1 in 4,000. So that means in the US you have 80,000 or so patients with RP, 150,000 more or less in Europe. Now it is caused by gene mutations, but gene therapies are not necessarily a good answer here because there are over 100 different genes that mutates leading to RP. So you would have to invent a lot of gene therapies in order to treat all patients here. There is one gene therapy approved, which is Luxturna for treatment of RPE65. All other patients need to wait. They have no treatment available to them, and they have to live with declining vision that does lead to complete blindness to most patients. Now, our approach is not depending on a genetic cause. This is pure cell therapy, and will be available for all patients, so that it's a huge opportunity. So our cells differentiate into functional photoreceptors. That's a big deal. You're born with all the photoreceptors you'll ever have. Your eye will not produce new ones. So cell therapies, this is really a beautiful area for cell therapy to be used. It's exactly the promise of cell therapy, is to do something that no other therapies can do. And creating new photoreceptors is definitely one of those. We can see them integrating into retinal layers in animal models. We see integration, and the integration may also help in terms of creating durable trophic support. Now, there's a broad potential of different indications where this could be useful, but we very much focus on retinitis pigmentosa for the moment. This was licensed in from Skepins Eye Research Institute, which is part of Harvard, some years back, and we still work very closely with them, as well as UCL and London. We spend a long time making sure that we can manufacture these cells correctly, and we have very good IP around that. The cryopreserved formulation gives 9-month shelf life currently, something that we obviously will expand over time, but 9-month shelf life is very much useful in order to be able to ship and store these cells. Now, it is a large orphan market, as I said. Luxturna has been approved. Luxturna is priced around $850,000 for 2 eyes, so they have set a kind of pricing precedent. We obviously have a lot more patients that are potential treatment candidates than Luxturna, since we're not gene specific. We've completed a phase 1 study in 12 patients, where we were able to establish safety in both fresh and cryopreserved cells, and we were able to upscale it up to 1 million cells as the dose, which we used for the first 10 patients in our RP study. And I'll show you those data in a second. Mass Eye and Ear in Boston, and the Retinal Research Institute in Phoenix were the 2 sites for those 2 studies. And before we go there, so this is the surgical technique. It is the same technique used by Luxturna, so subretinal surgery. It allows to place the cells in the right place, not only for integration, but also the right place for trophic effects. So in ophthalmology, you measure how many letters can be read, and typically you treat only one eye. You don't want to risk, if they have any safety concerns, you don't want to risk both eyes in one patient, and you treat the worst eye if they are different, and then you follow both. We have seen, so the top line here is the treated eye, and the bottom line is the untreated eye. And what you can see throughout the different time points, you can see here from month 1 out to month 12, that we have, it's hovering around 8 to 10 letter difference on the treated eye. And you can see the untreated eye. I mean, it really should be around 0 logically, but in small patient numbers, it will jump around a little bit. So, we're very happy to see a very consistent result over the time period. And if you hone in on 12 months, which will be the primary endpoint most likely in the phase 3 study, that's typically what the FDA will ask for. You can see that every single patient here of the 7 patients that reached that time point, who had a good surgical result, you can see that all of them have had some improvement. some really profound, 10 letters and above, and some less. I would say anyone from kind of an 8-letter impact upwards, it is really important impact. If you look at approval for Eylea, for example, Eylea was probably around 8 to 10 letters improvement. Obviously, different product, different disease, etc. But this is absolutely approvable efficacy rates. Put it that way. The mean here is plus 9.9 letters in the treated eye. Now, if you look at that, I guess you have a couple of conclusions. One is that this looks great, and let's multiply this by in a larger patient group and go for approval. The other thing we see is that we have no dose-limiting toxicities, and there is room for more, not much more, but we think we can double the dose in terms of space available under the retina. So we decided we'd like to see what happens if we double the dose, if we can get kind of the patients all the way on the left here to see if we can move them up even higher. And so that's what's ongoing now. We've done a phase 2A extension study where we're treating 9 additional subjects with RP, where we double the dose to 2 million. And there are a couple of other changes as well in the protocol, so we can look deeper at efficacy. And we're adding a couple of more sites. The KCI Institute has been added, and we're also opening one further site in Barcelona, and Oxford Eye Hospital at Oxford with Professor McLaren is also on the way to be open. So the next steps here for the retinal platform for us is to continue to collect data of the treated patients, as well as make sure that we get all the remaining patients in the high dose expansion study treated. So far, we have treated 3 of them, and we have 6 more that we're planning to treat. Then we would like to present that data later this year. There are some key conferences coming in Q3, Q4, so we'll aim for one of those conferences to present that update. And then following that will be to have a discussion with the FDA about how the phase 3 should look like. There is actually a briefing paper available on this, and it's pretty clear already what needs to be done. A single further clinical trial is what we expect to do. We expect that study to be randomized controls, but not placebo controls. In other words, patients will be randomized into either one of 2 treatment groups or into an observational cohort. But obviously the patient will know that they are not getting the good stuff, but that is it is impossible to do a proper placebo control of subretinal surgery, so there's precedent of doing it this way. Our strategy then will obviously, in terms of partnering, we will make a decision if we wanted to partner or go it alone, etc. after this data is available. More to follow later in 2021. Just a quick view of the therapy landscape. So there are really only 2 companies working in cell therapy in retinitis pigmentosa. It's ourselves and JSight. JSight has presented some data earlier, or in the middle of last year, in retinitis pigmentosa. The 2 big differences really is that JSight is using intravitreal injections, which is easier to do, but probably also gives less profound and durable efficacy, at least in our view. And they also do not have a cryopreserved formulation, at least not yet. In terms of other competitors, they all are working in gene therapy, and we discussed that a little bit earlier, so I have them listed here. Couple of words on the exosome platform. So exosomes is now a very hot field in research, and I'll skip what they are because I think that's getting more and more known. The advantages of our exosomes is that they are derived from a neural stem cell, and that may be the explanation why they seem to be favorably distributed across the blood-brain barrier. We've proven that we can load them with microRNAs, proteins, We have stable, consistent yields. We have well-established analytics, etc. So, and in addition to that, we're also engineering them to target other tissues other than the CNS. And our strategy here is a strategy of collaborations. We have significant research collaborations ongoing with large companies, all focused on delivering gene therapy. The way it works is the partner sends the cargo to us. We load that cargo into our exosomes, and we return it to them for further proof of concept studies. Should we have positive proof of concept here in one or more of these collaborations, that will then, we expect that to lead to a licensing deal with the collaborator. So I think there is a huge potential here for some proper value realization. JSAT has done a deal with Santen, which we think is a very credible deal, and that is for rights excluding the US, so non-US rights. On the exosome field, you can see the two large companies there, Kodiak and Evox, both have signed two important deals each. And they also have done some large fundraisers. Both of them, Kodiak with an IPO, and Evox did a Series C recently, raising almost $100 million. So, I would say both companies have shown that there is a lot of interest in exosomes and a lot of value there. So, in summary, there is a very strong news flow coming from ReNeuron over the next 12-18 months, both on the human retinal progenitor cells as well as exosomes. And there is another program I haven't talked about, which is iPSCs, where we also have a huge potential over the next few months. So thank you for your time and interest, and hope you have a continued great meeting. Thank you.